Hello and welcome to Cells in Chapter 1. So, when you think about cells as the basic units of living organisms, the interrelationships between cell structures shows how the cell functions to transfer energy, produce biological molecules including proteins, which are so very important in living things, and exchange substances with their surroundings. Prokaryotic cells and eukaryotic cells share some features, but the differences between them illustrate the divide between the two types of cells. Now, you guys have learned about prokaryotic cells and eukaryotic cells in STEM, but we will be talking about them and defining them again today. Okay, so that's your Cambridge objective, and we will be doing this in lab once lab starts. Okay, so an image you guys should be very familiar with. This is a picture of an animal and versus a plant cell. You need to be very familiar with the differences between these two cells. So we're going to be talking about in the next 3 videos. Okay, so I broke up the 80 slide presentation that I originally had for the cell into 3 different videos. So through these 3 different videos, we're going to be talking about most, if not all, of these structures that you're seeing, but you really need to put in the time to memorize each of these structures and how each of these cells are unique from each other. Okay, so if you're looking at through a microscope and you're looking at plant versus animal cells, so the main differences that you're going to see are in plant cells, you're going to see, you should see chloroplasts as well as cell walls. So you'll see microtubules, but really it's the cell wall and the chloroplast that are the 2 most defining characteristics. And that's how you know, without a doubt, that you're looking at a plant cell. Versus on the other side of the screen, you see animal cells, where you're not seeing these like defined cells within cell walls. Okay, so is this a plant or animal? And how do you know? Okay, it's an animal. What type of microscope is this? Okay, it is a compound light microscope. Okay, is this a plant or an animal, and how do you know? Okay, this is animal. How do you know? Because you're not seeing the really complex cell walls. And what type of microscope? It is a electron microscope. Okay, again here, what type of cell is this? It's plant. You can tell by that huge, large vacuole as well as the cell wall that you can see surrounding. What type of microscope? You guys should know. Scanning electron. Okay, and then this one, is it plant or animal? It's animal. You can tell by the types of cells that are there. You're not seeing those cell walls. And what type of microscope? This is a compound light microscope. Okay, plant or animal? Plant. Look at those beautifully defined cell walls. What type of microscope is this? It is a scanning electron. Okay, so here are the Cambridge objectives that you need to know. They are in your syllabus. I'm not going to spend a lot of time on this slide. Okay, so let's move on to talking about the features of an animal cell. Okay, first we'll start with the nucleus. The nucleus is the genetic library of the cell. The nucleus is the largest organelle that's in a cell. It's surrounded by 2 membranes known as the nuclear envelope. The outer membrane of the nuclear envelope is continuous with the endoplasmic reticulum. We'll talk about what that is. Okay, the nuclear envelope has many small pores called nuclear pores, which allow and control the exchange between the nucleus and the cytoplasm. Okay, so things are coming into the nucleus and things are coming out of the nucleus, like mRNA, okay, and we'll learn more about that when we talk about protein synthesis. Okay, so out is mRNA, ribosomes, and then in are proteins, nucleotides, ATP, and hormones. So there has to be some kind of system of control. When you have that many things coming in and out of the cell, it would seem like, or I'm sorry, not the cell, the organelle, it would seem like anything could get in or out, which obviously is not the case. So that membrane is really good at controlling what substances come in and out of the cell. Within the nucleus, chromosomes are in a loosely coiled state known as chromatin. So the chromatin condenses to form chromosomes during cell division. So when you think about that very typical image that you see of a chromosome that looks like an X, okay, that is when chromosomes are coiled and replicating. Really, though, in their sort of everyday, well, they're replicating every day, but when they're not in replication, they sort of look like a big loose plate of spaghetti. Okay, little strands that are loosely coiled, called chromatin. Okay, and also within the nucleus is the nucleolus, which is responsible for assembling ribosomes.
Welcome to Moo Math and Science. In this video, I'd like to look at the nucleus. I've always heard that the nucleus is the brain of the cell. But why is it the brain? And what are the major parts of the nucleus? The nucleus is a large organelle. In fact, you can see the nucleus with a compound microscope. Here it is inside a cheek cell. I'd like to look at four main components of the nucleus. First, the nuclear membrane, nuclear pores, nucleoplasm, and the nucleolus. The nucleus is surrounded by a nuclear membrane, and this is very similar to the cell membrane, and it is also composed of two layers. It protects and houses the other parts of the nucleus. You can also find ribosomes on the outside of the nuclear membrane. Up next are the nuclear pores. These are openings found on the nuclear membrane. They allow items to move in and out of the nucleus. An example would be messenger RNA.
You are heading through a nuclear pore and into the plant cell nucleus. The nucleus is home to a plant's DNA. Like meters and meters of cell blueprints, the DNA make the plant what it is.
Nucleoplasm is a gel-like or watery material found inside the nuclear membrane. You will find the DNA suspended in the nucleoplasm. The nucleolus is almost like a nucleus inside the nucleus. It has a very high concentration of DNA. It is also the site of transcription, which is the starting point of protein synthesis. This is how the nucleus controls the cell. It has all the information on how to make the stuff like proteins for the cell. These proteins are used for many purposes inside the cell. Ribosomes are also made at the nucleolus. All eukaryotic cells have a nucleus, and bacteria and other prokaryotic cells do not have a nucleus. Hope that helps.
The endomembrane system regulates protein traffic and performs metabolic functions in the cell. So it's an entire system. Components of it include the nuclear envelope, endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, and plasma membrane. So these components are either continuous or connected via transfer by vesicles. Okay, so again, the endomembrane system regulates protein traffic and performs metabolic functions in the cell.
The endomembrane system is a collection of membrane-based organelles that work together to create, modify, and export cell products such as proteins and lipids. It consists of the nuclear envelope, the rough and smooth endoplasmic reticulum, the Golgi apparatus, and several types of vesicles. Let's start our tour with the endoplasmic reticulum. There are two types of endoplasmic reticulum, rough and smooth. The rough endoplasmic reticulum, or rough ER, is studded with ribosomes. These ribosomes are what give the rough ER its rough appearance. Ribosomes are responsible for producing proteins, which are packaged in the transport vesicles and carried to the Golgi Apparatus. The smooth endoplasmic reticulum, or smooth ER, lacks ribosomes on its surface. This is why it's called smooth. The smooth ER is mainly responsible for the detoxification of harmful chemicals. It's also responsible for the production of lipids, which are also packaged in the transport vesicles and sent to the Golgi Apparatus. The Golgi Apparatus is made of a series of flattened sacs that look a little like pancakes. Its job is to package and distribute cell products that are either destined for a specific location within the cell, such as the plasma membrane, or for secretion from the cell. You can think of the Golgi Apparatus as the post office of the cell, where packages are dropped off and destinations are assigned. For example, some of the proteins from the rough ER are modified by the Golgi Apparatus to become digestive enzymes, which are packaged into a special type of vesicle called a lysosome. The job of this particular lysosome is to fuse with another vesicle coming in from the plasma membrane. The Lysosomes Digestive enzymes then break down the contents of the vesicle, releasing nutrients for the cell. Other cell materials leaving the Golgi Apparatus may be destined for secretion from the cell. For example, some might be hormones that help with body regulation, while others might be proteins that become part of the materials surrounding the cell. Before these products can be exported from the cell, they must first be packaged into a secretory vesicle. This vesicle then merges with the plasma membrane, releasing its contents into the external environment. The endomembrane system can be modified slightly to meet the specific needs of each cell. For example, a muscle cell usually has more rough ER because of its need for protein, while a liver cell usually contains more smooth ER because of its role in detoxification. Regardless of these modifications, the movement of materials within the endomembrane system is basically the same for all eukaryotic cells.
Okay, so as you just saw in the video, you can see a picture of the smooth ER connected to the rough ER. And again, the rough ER is connected to the nuclear envelope. You can see ribosomes there, the transport vesicle. Okay, so let's see. Okay, so transport vesicles moving from the rough ER into the Golgi apparatus. Now, you guys really need to know this process. So that video that I showed was a really good example. You really need to know what is moving, which, and we're going to go into more detail into some of these things, like we're going to learn about transcription and translation and protein synthesis in much more detail, but you need to know that mRNA is leaving the nucleus and that it's coming out and meeting a ribosome. You need to know what kinds of things are going into the rough ER, moving through the rough ER, being packaged, moving into the smooth ER, into the Golgi apparatus, what the transport vesicles do. It's a system, and you really need to know exactly, like that video showed, how that system works. Okay, and then, okay, so biosynthetic factories. The endoplasmic reticulum is a system of membranes running through the cytoplasm that is continuous with the outer membrane of the nuclear envelope. The ER accounts for more than half of the total membrane in all eukaryotic cells. Okay. Oops. Okay, so there are two main types of endoplasmic reticulum. There's the rough ER, and then there's the smooth ER. So the rough ER are covered with bound ribosomes, okay, ribosomes that are bound to the ER itself, and these are the sites of protein synthesis. So here, protein processing occurs, vesicle formation, who will be on their way to the Golgi, occurs, and it is itself a membrane factory for the cell. And then the smooth ER lacks those bound ribosomes, hence smooth. Okay, it synthesizes lipids and steroids. Okay, so it's synthesizing cholesterol, estrogen, testosterone, etc. This is also a place where poison is detoxed, carbohydrates are metabolized, and it can also be a place for the storage of calcium. Okay, so moving from the ER into the Golgi apparatus. Okay, Golgi apparatus is often thought of as the shipping and receiving part of the cell. The Golgi apparatus is a stack of flattened sacs called the cisternate and is sometimes referred to as the Golgi body. So there are more than one that can be present within a cell, and the main functions are to modify the products of the ER, manufacture certain macromolecules, sorts and packages materials into transport vesicles for transport either out of the cell or to other organelles within the cell. The stack is constantly being formed at one end from vesicles which bud off from the ER and broken down again at the other end to form Golgi vesicles. The Golgi apparatus collects, processes, and sorts molecules, particularly proteins, from the ER, and are made ready for transport in Golgi vesicles either to other parts of the cell or out of the cell. Okay. In plants, enzymes in the Golgi apparatus convert sugars into cell wall components. Golgi vesicles are used to make lysosomes, and it's involved in protein processing. So an example of that is an addition of sugars to proteins to make glycoproteins, etc. So protein processing. Okay, and so this French fry, you'll never look at Chick-fil-A French fries the same again, look just like a Golgi apparatus. Okay, so moving on to lysosomes. Lysosomes are the digestive compartments. Lysosomes are spherical sacs of hydrolytic enzymes surrounded by a single membrane and have no internal structures. Okay, they are very tiny, 0.1 to 0.5 micrometers in diameter. Their main functions are lysosomal enzymes can hydrolyze proteins, fats, polysaccharides, and nucleic acids. Okay, that's a huge function. Lysosomes also use enzymes to recycle organelles and macromolecules in a process called autophagy, and we'll talk about that more in a second. And periosomes are oxidative organelles. So they contain hydrolytic enzymes, which must be kept separate from the rest of the cell to prevent damage. So hydrolytic enzymes are enzymes that break down the components of a cell. And so this is used to recycle old dead things, viruses or bacteria that get in there that are harmful. There's all kinds of uses for hydrolytic enzymes, but if they were just floating around in the cell, they would be digesting the cell at large. So they have to be kept in these little sacs to prevent damage. And they're responsible for the breakdown or digestion of unwanted structures. In white blood cells, lysosomes are used to digest bacteria. Lysosomes sometimes release enzymes outside of the cell. And one really cool example, heads of sperm contain a very specialized lysosome called an acrosome for digesting a path to the ovum.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
Okay, so here is an image that is similar to the video you just watched. So in the top image, you can see some lysosomes in there. Okay, and then in the bottom image, you can see that lysosome contains active hydrolytic enzymes, okay, or digestive enzymes. And you can see a food vacuole that's coming into the cell. It fuses with the lysosome, and then the hydrolytic enzymes digest the food particles. Here is a wonderful microscope image at the top, a lysosome containing two damaged organelles. Okay, so you can see it's a fragment of a mitochondria and a fragment of a peroxisome that's in there that the lysosome is digesting. Okay, so the lysosome fuses with a vesicle containing a damaged organelle. and the hydrolytic enzymes digest the organelle's components. Okay, and this is called autophagy, which is lysosome breaking down damaged organelles. And then, before it's phagocytosis is the lysosome digesting food. So those are the two main functions of a lysosome. Phagocytosis, which is the lysosome digesting food, and autophagy, which is the lysosome breaking down a damaged organelle. Okay, cell surface membrane. So we are going to talk a lot more about cell surface membrane when we get to cell transport, but just as a part of the cell structure, we'll just talk about it in general for now. The cell surface membrane is extremely thin, okay, it's less than 7 nanometers. It has a trilaminar appearance at high magnifications. It is partially permeable, of course, because we know that things need to come in and out of that cell membrane. And it controls exchange between the cell and its environment, both of its internal environment and its external environment. And a lot of times you'll see it also called the plasma membrane. So in the literature, cell membrane and plasma membrane are the same thing. Okay, so looking at a picture here, this is the structure of a plasma membrane or a cell membrane, and this is how it is partially permeable. So there's a hydrophilic region, which we know hydrophilic region likes water. Okay, it is attracted to water, and we know it's those hydrogen bonds in there. That is, water is attracted to those components. And then hydrophobic region are areas that don't have hydrogen bonds and are not attracted to water. Okay, so you see there's a hydrophilic region of that membrane on the inside of the cell and a hydrophilic membrane on the outside, and then on the inside is lined with hydrophobic tails. Okay, and then lodged within that membrane are carbohydrate side chains and proteins, and these help to attract and control and allow things, bigger things like food particles, in and out of the cell. And again, we're going to go into much more detail about this in another chapter. Okay, the plasma membrane is a selective barrier that allows sufficient passage of things like oxygen, nutrients, and waste. The general structure of a biological membrane is double layer of phospholipids. And again, we will go into extreme detail in cell membranes and transport, which is chapter 4. Okay, microvilli. Microvilli are finger-like extensions of the CSM, which is the cell membrane. It is typical of certain epithelial cells, and what it does is almost always this structure, this like maze-looking structure, increases the surface area. Okay, and that helps because it helps absorption in the gut and reabsorption and proximal convoluted tubule in the kidney. So most places, like even in our brain, in our intestines, here in our cells, this like surface that looks at this back and forth extensions increases surface area. Okay, it saves space. That is the end of the cell part 1. You will find the video for cell part 2 next. Thank you.